Hi, I'm Mitra, and my lab studies regulation of ovarian cancer metastases by the microenvironment. And I'm going to give you a very quick summary of what we do. So, ovarian cancer is not the most common cancer, so it doesn't even list amongst the top 10 cancers amongst women, but it is the fifth leading cause for cancer-related deaths amongst women. And it's the most lethal gynecologic malignancy. So that's why we study ovarian cancer, and we specifically look at ovarian cancer metastases because 70% of patients who are diagnosed with ovarian cancer present with metastatic disease at the time of diagnosis. So they already have metastases, and we are treating metastatic ovarian cancer in the clinic all the time. But we do not understand the process of metastases that very well. And that's why it's very important to study it in order to design therapies that specifically target metastases. So ovarian cancer spreads predominantly within the peritoneal cavity, and it's very easy for it to spread because all the cancer cells have to do is that they exfoliate from the primary tumors, and then they are carried throughout the peritoneal cavity by the flow of the peritoneal fluid, and then they attach to the various organs within the peritoneal cavity. The most common site is the omentum, which is basically an 8-inch by 8-inch fat pad which hangs over the stomach and the bowels. And we mimic the omentum surface in the 3D culture model that we use in the lab. So basically we take omentum from patients, and then we isolate the mesothelial cells which cover the surface of the omentum, and the fibroblasts which form the basement membrane just below the mesothelial layer, and then grow them as primary cultures. And then we can use these to reconstitute in the petri dish the surface layers of the omentum. So we can form a basement membrane-like structure first and overlay that with mesothelial cells from the same patient to form a mesothelial layer. So these two together mimic the outer layers of the omentum. And then we can seed ovarian cancer cells with a fluorescent tag, and then follow what's happening in these cancer cells as they interact with the microenvironment of the metastases. So this is a section of actual human omentum. So this is the outer layer of mesothelium, and then there is the basement membrane, which has these fibroblasts. So we are mimicking exactly that in the tissue culture dish. So what happens during metastases, these cancer cells come and attach to the surface of the omentum, and then they invade through this mesothelial layer and then the basement membrane into the tissue parenchyma. And for successful metastases to happen, there has to be productive interactions between the cancer cells and the metastatic microenvironment that helps these cancer cells adapt to this new microenvironment. And we try to understand the crosstalk between the cancer cells and the new microenvironment and the adaptive responses that are induced. So how do we do that? So we study what happens when these cancer cells come and attach to the surface of the omentum. So this is the layer of mesothelium, and these are the fibroblasts. And this is the cancer cells which come and attach. So now what's the signals coming from this microenvironment which is received by these cancer cells? And what are the intracellular signaling pathways that are triggered as a result of these interactions? These can be paracrine, these can be juxtacrine. And then these signaling cascades that are activated can lead to changes in transcription factors and microRNAs. Both of them are involved in gene regulation. So gene regulation is important for inducing an adaptive response. So the cancer cells now need to either express or suppress certain genes so that they can thrive in this new microenvironment. So what are the altered targets of these microRNAs and transcription factors? And how does that lead to metastases, or can reprogram the normal cells in the microenvironment into cells which can support the cancer cells, so activated stroma. And in the third project, we study how this activated stroma, mainly cancer-associated fibroblasts, in the tumor microenvironment then can serve as a cancer stem cell niche for these cancer cells to then establish the metastases, and these cancer stem cells can also lead to chemoresistance as they survive the conventional chemotherapy regimens and then cause relapse of the tumor. So these are the main projects that we study. And our overall approach is that we base our findings on a combination of endpoint analysis of primary tumors compared to metastases in ovarian cancer patients. And then we also do 3D culture studies using our Omental 3D culture model, which mimics early colonization. Now, when we take those transcription factors or epigenetic regulators that are common to both, those would be factors that are changing really early during colonization and remain changed in the advanced metastases. So next we do functional analysis, follow that up with mechanism of action of those targets, and we end using in vivo preclinical validation in mouse models of ovarian cancer metastases. And we also test in patient data that is already available for prognostic indicators. So that basically summarizes what we do. Thank you.